Welcome, folks. And I have been asked to talk to you a little bit about the research that I do. Welcome here to my house. This is actually where I do all of my work nowadays in the era of COVID-19. It's otherwise known as what my kids call the Lizard Lounge. Go figure, I would hang out in a place called the Lizard Lounge. Well, anyway, let me tell you a little bit about the kind of work that I do, the kind of chemist I am, and then talk a little bit about a few of the projects that I do in my lab. So first of all, as you probably know, I'm an analytical chemist, and analytical chemistry is really the very applied side of chemistry. All right, so what we really focus on here is measurement in chemistry. So we're all about making good measurements in the chemical realm. All right, so how do we do this? Well, we can use very simple techniques such as wet analytical techniques or gravimetric techniques like we do in the quantitative analysis lab, which is a course I think most of you have taken. Or we can actually use very sophisticated instrumentation. So the kind of stuff that you learn about, for example, in my analytical course, and then of course access to other instrumentation that we don't even talk about there. So I do a large number of projects, very applied, as I mentioned, in my history at Shippensburg. I've done all sorts of stuff from theoretical to applied electrochemistry, sensor development, all kinds of stuff. I've even gotten into a little bit of biochemistry from time to time. So to be honest, I do pretty much a little bit of everything. So let me just tell you a little bit about three projects that I have going on right now that are sort of relevant to what I'm up to these days. So one recent project that I've worked on, but I haven't worked on for a couple of years, but could easily be brought back to the forefront if somebody were interested, would be analysis of materials that we get from the Shippensburg University Fashion Archives. So that's a collection of old garments and things like that that go back sometimes as far as the 1700s. So they're actually quite interesting to look at. So one project that we had a year or so ago that turned out to generate a considerable amount of interest was analysis of arsenic in 19th century women's garments. So back in the early to mid 1800s, people were very interested in developing bright, colorful dyes. And one of these dyes was a green dye. And this green actually contained arsenic. So of course that's harmful to anybody that comes into contact with it. So people who study these garments and collect these garments kind of want to know whether there's really arsenic in them or not. So we developed a test or a series of tests that allow us to look at those without being destructive. So obviously if you have a dress from 1820, you don't want to have to cut it up and analyze it. So using very small amounts, threads or very small patches of the garment, we actually developed an indirect analysis actually using copper, which was part of the dye along with the arsenic, to form a copper ammonia complex that absorbs light in the visible part of the spectrum. And then we can use simple visible spectroscopy to generate a standard curve and then determine based on a mass percent how much arsenic was actually in the garment. And then we also developed another method using atomic absorption spectrometry. And we actually did a little bit of atomic emission as well as a means of being able to make comparisons. So that turned out to be a very interesting project that we ended up presenting actually at a fashion and garments meeting, and it ended up generating a considerable amount of interest. So I also know that the director of the archives has a number of other types of samples that she would love us to actually analyze. So if you have interest in that, that's a project that we could definitely work on. A second project that we're initiating is ink analysis in potentially forged documents. So this sort of falls back into the forensics field somewhat. And here we're working with Doug Schaefer, who actually is a former analyst with the Secret Service. And you may have met him because I think last year he came and gave a presentation to the department. What we're interested in doing there is analyzing volatile components in inks on inked documents using gas chromatography mass spectrometry, GCMS. And the idea here is that we would like to be able to determine whether a document has been forged or not based upon the concentration or amount of specific volatile components that are found in specific inks. So we can, for example, look at a document, look for a specific volatile component in the ink using GC mass spec, and then determine whether or not the ink is consistent with the supposed age of the document. And if it isn't, then we probably have a forgery on our hands. So we have a number of characterizations that we'd like to do there with Mr. Schaefer. Things, for example, like how much does heat affect these measurements? How much would humidity affect these measurements? So this is sort of a new area of forensic science, and turns out Doug is pretty much one of the forefront leaders in that area, and is very interested in working with us. So that's another project that we potentially have going that has, I think, a lot of excitement in the forensic side. And then another very applied thing that we've been doing recently is working on analysis and quality control of kombucha. Now, kombucha is a tea-based fermented beverage. You may or may not have tried it. It's actually quite refreshing and quite good, and I'll admit I'm somewhat addicted to it. And we're actually working now with a local small batch kombucha manufacturer, and we're helping that business to actually develop a quality control protocol so that they can actually improve the quality control of their different products. So as a result, we end up doing analysis of kombucha. It's very simple things. For example, like pH, we do titrations like in Quant Lab, so we can titrate total amount of organic acids that are in kombucha that give the kombucha its flavor. We look at sugar content through refractive index, and most interestingly, we're doing a lot of work with alcohol content. And the reason for this is because to sell kombucha as a non-alcoholic beverage, you have to keep the alcohol content below a certain value. So it's very important from a quality control perspective to be able to show that we can do that. So we're actually comparing 3 different methods of alcohol analysis, from the quite inexpensive all the way up to the fairly expensive, to see which ones we can get away with. And there's a lot of really interesting science really associated with that alcohol analysis. It's kind of a difficult thing to do, but I'm finding it actually to be somewhat fascinating. And then ultimately what we'd like to be able to do is tie, for example, the alcohol content back into the brewing process itself, and understand, for example, whether type of tea, the amount of sugar you use, the brewing time, the brewing temperature, all of these things are going to affect the alcohol content. So we can use that to sort of help us to drive the brewing process. So that's another very interesting sort of food chemistry application we're working on. And then finally, we've had in the past and continue to have a number of different collaborative projects with Dr. Kagaris. They can fall into one of 2 categories. One of these would be taking, for example, an interesting set of experiments that we do as part of our research, and then adapting that into some sort of a laboratory exercise that you could apply to, say, analytical chemistry or PChem or even general chemistry. So for example, we're now developing an experiment based around the alcohol analysis in the kombucha that I just talked about, that we could actually use in the quantitative analysis lab or even in the dynamics lab. And then the other thing that we do is we look at other areas such as sensor development and materials, and my group comes up with interesting measurements that we can do using instrumentation. And then Dr. Kagaris's role is to help us to develop mathematical models that can then be used to help us to better explain the data that we're seeing. So as you can see, we have a number of different applied and somewhat theoretical things going on in my lab. So if you're interested, of course, feel free to talk to me, and hopefully we'll get to see you again soon in the fall.